Assalamu alaikum dear students. Today we will discuss PCR polymerase chain reaction MCQs part 1A. A small piece of DNA can be amplified by option A PCR, B denaturation, C hybridization. Correct option is A PCR polymerase chain reaction. An initiation DNA is made of option A double stranded, B spiral shaped, C single stranded, and correct option is C single stranded. Subscribe now and press the bell icon. Never miss an update from a technologist. PCR is generally option A 4-step reaction, B 2-step reaction, C 3-step reaction, and option D 5-step reaction. Correct option is C 3-step reaction. For gene transfer to be effective, transforming DNA must be option A incorporated into the bacterial chromosome, B incorporated into a viral genome, C free in the bacterial cytoplasm. Correct option is A incorporated into the bacterial chromosome. The viruses that can achieve neoplastic transformation are called option A DNA tumor viruses, B RNA tumor viruses, C retroviruses HIV, and option D none of these. So the correct option is A DNA tumor viruses. From a single molecule of DNA, PCR can make option A one additional copy, B hundreds of copies, C thousands of copies, option D millions of copies. So the correct option is D millions of copies. Double stranded DNA denaturation with a specified limit of temperature is option A reversible reaction, B irreversible reaction, C either A or B, and option D none of these. Correct option is A reversible reaction. Specialized transduction occurs when option A the bacteriophage incorporates randomly into the bacterial chromosome, B the bacteriophage never incorporates into the bacterial chromosome, option C the bacteriophage always incorporates at the same position in the bacterial chromosome, and option D none of the above. Correct option is C. The bacteriophage always incorporates at the same position in the bacterial chromosome. Bacteriophages are option A cells in the blood that eat bacteria, B a class of bacteria, C bacterial viruses, D none of these. Correct option is C bacterial viruses. PCR can be used to amplify a specific fragment of DNA. Which of the following can be used? Option A drop of blood, B a hair follicle, C a fragment of skin, D all of these. So the correct option is D all of these. Transformation means option A formation of pills, B acquiring DNA from the bacterial cell environment, C plasmid containing factors. Correct option is B acquiring DNA from the bacterial cell environment. Bacteriophages are option A cells in the blood that eat bacteria, B class of bacteria, C bacterial viruses, option D none of the above. Correct option is C bacterial viruses. Restriction maps of option A allows comparison between DNA molecules without the need to determine nucleotide sequence. B allows comparison between DNA molecules but require determining the nucleotide sequence. C does not allow comparison between DNA molecules. Correct option is A. Allow comparison between DNA molecules without the need to determine nucleotide sequence. RNA is copied into complementary DNA, cDNA by option A. Take DNA polymerase, B. RNA polymerase 2, C. Reverse transcriptase, option D. Uracil N glycosylase. Correct option is C. Reverse transcriptase. A PCR cycle consists of option A. Denaturation, primary annealing and elongation. B. Denaturation, initiation and elongation. C. Primary annealing, elongation and termination. So correct option is A. Denaturation, primary annealing and elongation. Which of the following is an advantage of nested PCR? Option A. Typically has high sensitivity and specificity. B. Second round PCR products can be a source of laboratory contamination. C. It is less time consuming than single round conventional PCR. So correct option is A. Typically has high sensitivity and specificity. The reverse transcriptase reaction can be primed by option A. Target sequence specific primer. B. Random hexamer. C. Oligos DT primers. And option D. All of the above. So the correct option is D. All of the above. A plasmid encoding a target sequence of interest will be used as a quantitative PCR standard. Where should you work with the plasmid? Option A. The PCR reagent clean area. B. A special area designated for high risk templates. C. The area where all sample templates are prepared. Option D. None of the above. Correct option is B. A special area designated for high risk templates. The cycle threshold is option A. The total number of cycles performed during a real-time PCR reaction. B. The cycle that a sample crosses a certain point during a real-time PCR reaction. C. The cycle number at which a sample enters the plateau phase of PCR. Correct option is B. The cycle that a sample crosses a certain point during a real-time PCR reaction. Which is not a property of real-time PCR assay? Option A. Incorporate dyes that bind double-stranded DNA. B. Incorporate an internal hydrolysis probe. C. It can be performed at a single temperature with no specialized instrumentation required. Correct option is C. It can be performed at a single temperature with no specialized instrumentation required. Which of the following is not an advantage of quantitative PCR? qPCR. Option A. Reliable indicator of viable infection. B. No post PCR handling of products. C. Highly sensitive, specific and repeatable. Option D. Can obtain quantitative results. So the correct option is A, a reliable indicator of viable infection. Which of the following statements is false? Option A, PCR inhibitors can lead to false negative results. B, PCR examines a large proportion of the tissue, leading to false positive results. C, pathogens diversity at primer sites may lead to the false negative results. Option D, contamination may lead to false positive results. Correct option is B. PCR examine a large proportion of the tissue, leading to false positive result. Ruggedness is defined as reproducibility of an assay using different reagent brands or batches and different equipment. Option B, the minimum number of copies reliably detected by the assay. C, agreement between sample replicates both within an assay run and between independent assay run when tested by the same laboratory. Correct option is A. Reproducibility of an assay using different reagent brand or batches and different equipments. The limit of detection is synonymous with option A, repeatability, B, analytical specificity, C, analytical sensitivity. Correct option is C, analytical sensitivity. Various samples that ensure the validity of positive and negative results are called option A, controls, B, standards, C, exogenous normalizing variables, D, endogenous normalizing variables. Correct option is B, standards. The multi-stage process that evaluates an assay fit for its intended purpose is called option A, validation, B, QA or QC. Option C, accreditation, and option D, none of the above. So correct option is A, validation. Analytical specificity is defined as option A, the degree to which the assay does not detect, amplify other pathogen. B, the intended purpose of the assay. C, agreement between sample replicates both within an assay run and between independent assay runs when tested by the same laboratory. Correct option is A, the degree to which the assay does not detect, amplify other pathogens. A PCR reaction that contains only one copy of the target sequence, one copy per reaction, option A, is typically amplified in a highly repeatable manner. B, may amplify, but detection is not likely to be highly repeatable. C, can be precisely and accurately quantified using quantitative PCR. So the correct option is B, may amplify, but detection is not likely to be highly repeatable. Samples of known concentration per copy number used to construct a standard curve are called option A, controls, B, standards, C, exogenous normalizing variables, D, endogenous normalizing variables. Correct option is A, controls. Tissue weight would be an example of option A, control, B. Standard. C. Exogenous normalizing variable. D. Endogenous normalizing variable. Correct option is B. Standard. Which of the following is not a method for stabilizing RNA? Option A. 95% ethanol. B. Liquid nitrogen. C. RNA later. Correct option is A. 95% ethanol. Expression of a housekeeping gene would be an example of option A. Control. B. Standard. C. Exogenous normalizing variable. D. Endogenous normalizing variable. Correct option is B. Standard. Which of the following is correct? Option A. HIV is retrovirus that kills human helper T cells. B. Causes acquired immune deficiency syndrome AIDS. C. All of the above. Correct option is C. All of the above. Which of the following is not a stage of PCR? Option A. Decay. B. Platy. C. Lag. And option D. Exponential. Correct option is A. Decay. Thank you for watching my videos.